Hello, everyone. My name is Jihan Abdurrahman, and I'm going to talk today in my presentation about stem cell. I'm using stem cell to regenerate aging. So, aging is my concern today because I'm talking about aging as a complex biological process characterized by cellular decline and tissue dysfunction. To overcome this biological process, we need such a kind of stem cells. Why we pick up the stem cell? Because it has ability to differentiate into different kind of cell. So basically, the stem cell is undifferentiated cell, and we can use it in different kind of cell type. This has led me to my objective today. My objectives today, I have three categories of objectives. First of all, to recognize the different types of stem cell. The second one, to understand the potential of a stem cell in anti-aging process. And the third one, what are the techniques that I'm using and the equipment to help us using this kind of stem cell. So first of all, what is a stem cell? A stem cell is, as I said before, is undifferentiated cell with remarkable ability to self-renew, so it divides by cell division and replicate itself to maintain a pool of stem cell and to differentiate into different kind of stem cell like muscles, bone cell, nerve cell. So how can we obtain this kind of stem cell? We can obtain this kind of stem cell by two ways. First of all, when a mother delivers the baby, we have umbilical cord. Inside this umbilical cord, we have uh, an, a many or abundant of stem cell in this part, this rope. Um, this cell we can preserve it for a baby later in a cell bank. So this is the first way to obtain the stem cell. The second way, if we cannot catch this stage of the baby, so we can use it when the person become adult, and we can take it from the bone marrow. So from bone marrow, we can induce a biopsy to the bone marrow, and we can extract this kind of stem cell, and we put it in a culture, and also we keep it in a stem cell bank under the name of patient. So we don't have one kind of stem cell. We have different kind of stem cells. This has led us to know what are the types of stem cells. We have four types of stem cell. First type is totipotent stem cell. This is only found in a fertilized egg or in an embryo, but we cannot use it, unfortunately, this kind to use in anti-aging. Why? Because it's still in a zygote, so we don't have enough number of stem cells to help in treatment. Also not used in anti-aging research due to ethical concern surround the embryo destruction. So we don't need to destroy the embryo, so or make a portion. So this is kind of ethically issue. The second type of stem cell is pluripotent, and this is our concern today. And this is exists in a blastocyte stage of embryonic development. So when the cell divide into two cells and 4, 8, 16, until reach 32 cells, this is called blastocyte. It is just like an empty ball. Inside this empty ball, we can find the cell differentiate into three germ layer, like ectoderm, which is responsible to form skin, and mesoderm form the blood and the muscle, and the endoderm which form all the reproductive system and all the system inside the endoderm layer. So two main types source of embryonic cell, as we say, the embryonic or the bone marrow. The type of stem cell, the third type of stem cell is multipotent. Multipotent, this is found in all adults. And as we say, this is differentiate into bone marrow or adipose tissue like fat tissue or and umbilical cord blood. It differentiates into a limited number of cell type, like bone marrow stem cells can become bone cartilage or fat cells. This is about the multipotent stem cell. The fourth kind of stem cell, oligopotent stem cells, can differentiate into few closely related cell type with their lineage. Example for that, all the blood cells, all different kind of blood and brain cells. In this table, we just make a summary for the different kind of cell like totipotent, pluripotent, multipotent, and the last one is oligopotent. Um, and the potency, the origin, and the example of each one. Now let's talk about the techniques of the techniques used in a stem cell. So the technique that we can use in a stem cell, we can use two different types of techniques. We can use something called regenerative therapy. Regenerative therapy, this is by replacing the damaged or dysfunction tissue with a healthy stem cell, which drive replacement. This is being investigated for conditions like osteoarthritis and heart disease and muscular degeneration. So we can use this kind of stem cell to regenerate, for example, the cartilage. If someone suffer from the cartilage problem or make osteoarthritis in their knee, in his knee or her knee, what's happened? So we can take this kind of stem cell and we start to inject this stem cell in this part, the pain part, whether cartilage, with the or knee or any part of the body, to help this part to form a new kind of cell instead of dysfunction one or the damaged part. So this is help a lot in in the new medication field. The second types of technique, we can use cellular rejuvenation by stimulate existing cells to function more youthfully with factors secreted by stem cells. This approach is still under research and it's early stage, but hold potential for addressing aging-related decline at cellular level. Concerning the equipment that we can use in this process, we can use different equipment like incubator and bioreactor and other tools for maintaining and expanding the stem cell population into controlled equipment. First, we can use incubator because if we need the specific temperature, we need a suitable condition to keep and preserve this stem cells. The second equipment we can use is gene editing tools. This technique like CRISPR-Cas9, which are being explored to modify the gene in stem cells to correct the age-related mutation or enhance their therapeutic potential. So such a kind of technique, we can use it for genetic issues. But the last one which is bioprinting technology. This building tissue and organs, we can build up tissue or organs layers by layers. So we can build up layer by layer by using this stem cell and biomaterial with potential application in personalized medicine and organ uh, transplanting. Like when they make a face transplanting or something, or organ transplanting, like in the kidney or something like that. So we can use this bioprinting technology. What is the critical information on stem cell anti-aging? Of course, we can put in our consider the regenerative potential and the cellular regenerative and personalized medicine. All of this, the stem cell has ability to differentiate into diverse cell type and to replace the damaged and dysfunction cell. I think we talked about that before, but we talk about each one in detail, more details, and cell rejuvenation uh, to enhance the function of each cell and improving overall cellular health. That like they can use it in a cosmetic when they make a face rejuvenation. And the last one is personalized medicine. We can use the patient-derived stem cell allowed to for personalized therapy, targeting their specific genetic and cellular makeup. And this is a good example for how the cell induced in the different kind of cell. So we induce the pluripotent stem cells from adult and reprogram this pluripotent stem cell to an embryonic-like state, and we offer approach to generating tissue types specific to the individual, reducing rejection risk, and research on induced pluripotent stem cell in age-related condition like uh, Parkinson's disease is ongoing. Sometimes we can find side effect like tissue rejection when the stem cell possesses anti-inflammation property. They secreted factor that suppress inflammation. So we can put or give the patient some medication like cortisone or cortisol to make anti-inflammation for the tissue rejection at this time. Um, we can study suggested they could help manage chronic inflammatory condition linked to aging. So this is the last thing, which is the side effect that affect with the stem cells or can cause by stem cell. Um, and this is the in this picture we can find how can we grow the isolated cell from patient to the dish, omega fibroblast, and then we treatment the cell with reprogramming factor, and then we can wait for a few weeks to keep the cell undergoes the cell division. Then the pluripotent stem cell become ready at this time. So we have a stimulated cell to differentiate into varieties, um, variety kind of cells like cardiac, like here in the also in blood cells or even in a gut cells. If someone has ulcer or has a problem in the small intestine, so we can stimulate and induce this kind of pluripotent stem cells into these parts to healing. Thank you so much for watch, and this is the different resource that I have used. This is a general resource, this is a research article, and this is other resources in addition to the book, the fifth edition book for Dr. Wall. So thank you so much, and I hope this presentation got benefit for all. Thank you so much.